Okay, so we should restart, and the next talk is going to be by Bakhtier Vasiev, and he's speaking about modeling chemotactic motion of cells in biological tissues.
Hello, everyone. Thanks to organizers for inviting me here to the talk. Yeah. Okay, so I will be talking about modeling chemotactic motion. Concerning like bacteria or amoeba, it is pretty much clear how chemotactic motion takes place, but it's a bit more sophisticated when it comes to motion of cells in tissues. So this movie refused to play. I will show another movie very similar. So here is it's movie made in his virus lab. It's regression of primitive streak. There is a group of cells somewhere here. It's called stem zone. It contains around 1,000 cells, and this group of cells moves backward to posterior end. And there is some more motion on epithelium. So idea basically about primitive streak progression and primitive streak regression, one of ideas, one of hypotheses, that all motion in epiblast is orchestrated by moving group of cells. There is a single group of cells which moves along midline of embryo. So when progression takes place, they move toward anterior side. And when regression takes place, it goes toward posterior end of embryo. And if we assume that this is a K event during gastrulation, how we could model it? So this is what I told. During primitive streak progression, there is a, we can say, small group of cells which later forms Hansen's knot. It's around 80 cells. They move straight along midline of embryo. And here, a bit larger group of cells, stem zone, moves backward on the later stage of gastrulation. So these two images from computer simulations, I will talk about them a bit later. So assume there is a group of cells which moves in a tissue, and assume that this is motion due to chemotaxis. So the question is, what kind of chemotaxic scenario could be involved in this? For example, what cells produce chemotactic agent, what cells react, and if the reaction is it attraction or repulsion. If we consider just group of cells moving in a tissue, it looks like we have two cell types. Is two cell types enough to get this group of cells moving or not? And next question, if it's heterogeneous, then what kind of interactions can result to motion of this group of cells? So this idea comes about this group of cells is from these experiments about expression of FGF8 matrix RNA. So expression area is very small. This is image during time of regression of primitive streak. There is a group of cells. Let's say this is stem zone. In stem zone, there is active transcription of FGF8, but FGF8 proteins or matrix RNA, they are observed in large areas. So there is some kind of diffusion. And this is what we will assume in the model. So we will consider a group of cells. So in the first step, we assume that there is some protein produced in this group of cells, and it decays and diffuses everywhere. So this group of cells could be isolated, like here, or it could be inside of tissue. So then we can, so two-dimensional problems is still complicated problems. We can reduce it to one-dimensional problem. What we do, we draw a line which crosses this moving group of cells. For example, we assume that it moves horizontally, so the line should be horizontally in direction of motion. And then we can replace all this group of cells by a segment of line. And then moving group of cells will be replaced by moving segment. And this way we get one-dimensional model. In one-dimensional model, so we assume again, so there is a production of some kind of morphogen inside of this group. Let's concentration is denoted by symbol U. And then we will have equation for this concentration. This is diffusion, decay, which takes place everywhere, and production, which takes place only inside of this segment, in this segment. And if segment is moving, then we can jump into co-moving frame of reference. The equation gets slightly different. At one hand, it gets a bit more complicated because we get extra term, advective term. On the other hand, it gets simpler because now our segment is stationary. So it's easier to represent production term. For this, we can find analytical solution for this kind of problem. So if segment is not moving, we will have some concentration profile, symmetric concentration profile for our morphogen. If segment is moving, then it will be asymmetric. It will be slightly shift backward, yeah, due to motion. So in both cases, this is analytical solution. So if C is 0, we will have this profile symmetric. If C is non-zero, we will have this C positive. It will be backward transition of profile. If C is negative, it moves this way. It will be just symmetrical. Okay, and now as a next step, we assume that this motion of this migrating group of cells is due to chemotaxis. If it's due to chemotaxis, then we can, if we look here, we see that there is a, for example, if this segment moves to right, then concentration of this chemical is lower on the front and higher on the back. So this, if we want to say it is chemotaxis, then we can assume that it is repulsion. Anyway, so if U is chemotactic agent, then we can say that speed of motion is proportional. This is strength of chemotactic reaction, C naught, proportional to the average gradient over this segment. Let's take this assumption. This is it's this assumption made in many models. It started from Keller-Segal model. Anyway, but now we have, since we have a profile, we can find expressions for concentration of U at both sides of this segment, and therefore this equation transfers into this. There is a bit complicated right-hand side, but left-hand side is just C. So we can represent it as C equals to some function. So this it simplifies because this function will be different for different models, but we will always have C equals to some function of C.
Can you remind me what is lambda 1?
Lambda 1 and lambda 2, it should be a superposition of two exponents. If it's linear piecewise function, yeah, and this is exponentiations here. Now, so again, C equals to f of C. C is straight line, this line. F of C, it is one of these curves. These curves are different, so what I did here, for different values of C naught, for different values of chemotactic strength of reaction, we will have different profiles. Well, it's just stretching, it's proportional. So for repulsion will be these solid lines, for attraction this will be dashed lines, and now solutions of this C equals to f of C are points of intersection of this straight line and this one of these lines, which corresponds to given value of C naught, to given strength of chemotaxis. Now what we can see that if chemotaxis is weak, there is only one point of intersection. But if it's strong, we get two extra points of intersections. This point of intersection corresponds to motion to the right, and another one, motion to the left, which is reasonable because it's a symmetric problem. It could move either direction. And question appears why we have when we have this let's say, propagating wave solutions. When we have solutions, when this group of cells is moving, it can be this solution when C equals to 0 always exists. So it can be always stationary. But if C naught is high enough, if chemotactic is strong enough, it can start to move. And this is represented like it's shown here. It is basically pitchfork bifurcation. So this is, so C naught negative corresponds to repulsion, positive to attraction. When it's positive, we have only one solution, and this solution is stable. When C gets certain, achieves certain threshold value, then this stationary group of cells becomes unstable, and there appear two other solutions with non-zero speed of motion. So this is how it works, what it says. It says, now we can transfer it into language in biology. There is a group of cells. If this group of cells produce some chemical and reacts to the same chemical, chemotactically repelled by this chemical, and if this repulsion is strong enough, this group of cells will move. It can move rightwise or leftwise, but it will move. Similar, everything exactly the same if we assume that there is a chemical which is produced outside. There is a chemical which is produced outside, then it will be this kind of profile. Very high level of this U outside of this group of cells. Inside, due to decay, it will be lower. And then if it moves, we will have this difference between values of concentrations on the left and on the right. And the only difference now, this chemical should be attractant. So if outside cells produce chemical and this chemical is attractant for cells forming this group of cells, it will be also moving, and it's everything the same conditions. If we keep same parameters, everything will be the same. So it's the same solution. So here are some illustrations. This is moving segment or moving group of cells in 1D or in laboratory frame of reference. This is in co-moving frame of reference. So we just move together with this group of cells. Now this is, so here it was moving because we just imposed some kind of motion, and here it will be motion due to chemotaxis. So here chemical is produced inside, so it stays. We need to disturb slightly to get motion. So it stays. Since everything is symmetric, it will be, it's unstable position, unstable solution, but it will stay unless we perturb it. And this we don't need to perturb because asymmetry provided by boundary conditions, when it's close to boundary, will have slight difference in concentrations on the left and on the back, and then we can see that both boundaries repel this group of cells. But, well, it repels itself, but it will move, it will be reflected from both boundaries. And this is same mechanisms now in cellular pods model. So we have a group of cells which is repelling itself. So I did, when it repels itself, it can move in random direction. So it's unstable position, and it will move in random direction. Just to make sure that it will move in certain direction, I slightly play with initial conditions, slightly shift concentration profile to the left to make sure that this group will be moving to the right. It is same movie, but here I removed surrounding cells. It just shows concentration profile of U and moving group of cell. This is a second scenario when chemical is produced outside and it's attractant for this group of cells. And it is same movie. So this dark means high concentration. So concentration is lower inside of this group of cells because there is no production but only decay. Yeah.
Short question. I can imagine that once if the cells would happen to detach from one another, they will be chased away from one another.
Yeah, if they detach, then they will move a separated group of cells. So I tried to make strong enough. Well, we can say it's single cell. Yeah, if it's single cell, there are no problems. If there is a group of cells, then it should be strong adhesive contacts between them to stay together. When I look at larger group of cells, they make very strange shapes, which can't be modeled in 1D model. But it's again, so if it's not too large, it will stay connected. If it detaches, then two groups just move. Okay. So in both these scenarios, we chemotactically reacting cells were cells which move. In model which we just imposed, we can't consider chemotactic reaction of surrounding cells because surrounding medium has roughly speaking, infinite size. And we can look into this formula. So speed is some expression divided by size. So if size is increasing, then speed will reduce. Therefore, in infinite, let's put it like this, in infinite medium, we can't have motion of this group of cells if chemotactically reacting cells outside. Or we can put it a different way. We can say if this group of cells is increasing in size, then it should slow down. And to avoid these problems, we can slightly modify model. We can say that speed of motion is proportional not to average gradient over this group of cells, but proportional to the difference of concentrations on two sides. It's like if cell elongates, it feels better signal, but gradient is the same. Yeah, so size can make difference. So bigger cells can detect weaker signal. So if we put it like this, then we indeed will have we can have motion of large group of cells, and then increase of size results to increase of speed, but it saturates like here. So this is for small. We can see this part, this small part of increasing part. It's shown here, and here it's also this increasing part for original model when chemotactic and speed is proportional to the average gradient. So it's again, it's increasing with the size of the group of cells, but then when size is large enough, it starts to decline. So this is declination, and here it's slightly different in this model. I have checked for curiosity what happens in solar pots model, and they move. I don't know what of these two mechanisms really working in solar pots model. It looks at second one, because if it's first one, if I increase medium size, speed should decrease, speed of this moving group of cell. Yeah, roughly speaking. So A here would rather represent maybe not clear. It doesn't matter. So what I found that when I increase medium size, velocity is not changing. So this probably like this. So probably it's a second mechanism. But it's pointless to discuss what is in solar pots model because it has nothing to do with what is in real cells. And one more mechanism. I didn't show, I don't have this movie, but it is like counterpart. So every chemotactic mechanism comes with its counterpart. So this counterpart for this, cells which make a group, this group, they produce chemical, and this chemical is repellent for surrounding cells. In this case is identical in a sense to this case, which is in here. Like previous two cases were identical, these two cases also identical. And it's easy to see, so in solar pots model I have same speeds, everything is the same. Okay, we can slightly modify model now. If what happens if this moving group of cells is heterogeneous? In a sense, it's a bit easier because with heterogeneous group of cells, it's easy to see it will move. But same analytics can be used to find out what is, well, now there is no we can't talk about conditions for motion because it will always move. For example, in this case, we have group of cells. Half of this group is producing chemotactic agent, and half is reacting. Doesn't matter. It's attracting or repelling. So in both, so it will be moving either one or another direction. There is some asymmetry. If so, for example, if it's repulsion, eventually it's faster. So this group of cells eventually will move faster. If cells are repelled, it will be moving a bit slower. If it's attracted, it's because of shift of all this group with respect to this profile. So profile is on one side. If it's shifted towards this spike, difference between concentrations on two sides of this group gets a bit smaller. If it moves on the other side, it gets larger. So this gradient is different, and therefore there will be some difference in speed. But it will always move. It's opposite to what was this homogeneous group of cells. This homogeneous group of cells, there is always minimal conditions satisfied before it will move. So here also illustrations using cellular pods model. Here in first movie, blue cells attract green cells. Doesn't play. This play, okay, it's pretty much similar. Here blue cells repel green cells. It moves in, so it's pretty much, it's again, as I said, every chemotactic mechanism comes with its counterpart, but in 1D case they are identical. In 2D case they are different because shape of moving group of cells eventually is different for these two scenarios. I can't show it here, but I will.
Why are the blue cells moving?
Because they are attached to green cells.
Oh,
Yeah, that's again, so it's the same. So I make sure that this group of cells altogether is heterogeneous group of cells, but they don't split. So if they split, then it will stop.
What is the initial configuration?
Initial configuration, what it was here. So like doesn't not so important. You can make some group of cells connected and divided. So I divided 50-50, but you don't need even to divide 50-50. So there should be some one side.
Could you do this randomly and then they reorganize, or maybe not?
I don't know.
Or probably form small clusters of cell pairs.
They might, yeah.
Yeah.
So if they originally dispersed, can they sort out? I never checked it. It's interesting, yeah. But maybe I will check it later. Okay, and this is second case for moving heterogeneous group of cells. Now what we have is, it's again, some part, well, let's put it like this. Some cells are chemotactically reacting, some cells are chemotactically producing chemotactic agent, but this group of cells overlap. For example, whole group is reacting chemotactically, and only some subpart is producing this chemical, or other way around. Whole group is producing chemical, and only subpart is reacting. And so in middle case, it's what we considered as first case, when these two subgroups coincide. When these two subgroups coincide, we have this pitchfork bifurcation, but in these two other cases, we will have fold bifurcation. What is interesting, this interesting part here is that for some value of C0, if C0 is strong enough, we will have motion in both directions. So it can move to the right and it could move to the left. If it, well, I mean, in one direction it can move only with a speed above something. It's hard to see it here. It will be easier to see on this. It is in a sense it's similar. It's a second case for moving heterogeneous group of cells. Now what we have. So there is a group of cells. There is a production of chemical outside, let's say, and only part of this group of cells is chemotactically reacting. Yeah, so it's different from previous two cases. Then what we would expect? We would expect that if, for example, this reacting part on the left, and it is repulsion, it will move to the left. Yeah. But what happens if it can also move to the right? Can you see why? So we have a here.
Just, yes.
So this is our profile. This is our group of cells. For example, it moves in this direction. So it can move in this direction, and this will be repulsion. But it also can move repulsion. So if it moves here, it will be attraction, sorry. Then attraction. So here, if it on this side, it will move opposite direction. That's what I wanted to say. Yeah. If our group of cells located on this branch, it will move this direction. If it's on this branch, it will be moving this direction. But in both cases, we have attraction. So what I want, but in order for this to appear on the other side, it should move strong enough. So if it can't move, so it can move in one direction with any speed, on the other direction with speed above something. To make so this speed should make sure that it's on the other branch. Yeah, and the same story was in here also. This motion and this motion. This motion in the opposite direction, but eventually it comes to the same story. Okay. And here illustrations using solar pots model. So blue cells attract red cells. These blue cells attract red cells, and the red cells push them. And here, red cells repel blue cells. It's called, as I said, every chemotactic mechanism comes with its counterpart. What's it? So red cells repel blue cells, and this group of cells moves from the interface between red and blue cells.
Green cells doing here.
It makes sure that we have some kind of asymmetry. Without this asymmetry, we will have one of scenarios with homogeneous group of cells. So what makes this asymmetry? Asymmetry makes everything easier. When we don't have asymmetry, then, for example, chemotaxis should be very strong to get moving. But when we have asymmetry, then there are no limitations. If chemotaxis is very, very weak, it will still move, but very, very slow. So here green cells repel red cells. So interface between red and green moves towards red, and here red cells attract green cells. Same result. Interface. Well, it doesn't move, but it's pretty much same kind of story. Okay, and now it's about application of these mechanisms to formation of the primitive straight. So this image, these movies from paper we published with Kiswire. So now this it's more related to motion of heterogeneous group of cells. What we have here we should have at least 3 cell types. In here, yellow cells are attracted by chemical produced by red cells. So this white area means high concentration of this chemical. So we can see formation of primitive strake. So here it's again, since yellow cells attracted by red cells, interface between yellow and red will move towards red. And if blue cells are again attached strongly to yellow cells, we will have this formation of primitive strake. This is counterpart. When red cells are repelled by yellow cells, it's still the same. So this interface should move in the same direction. And again, we will have same kind of pattern. Okay, so here it's again white area, it's area where high concentration of this chemical, and here it is trajectories of some of cells. This is another mechanism which eventually produces the same. Here, blue cells repel yellow cells. So there is a chemical produced by blue cells. If they repel yellow cells, then interface between yellow and blue moves toward yellow, and this way we also have formation of primitive strake in the way it's shown here at least. And counterpart for this would be what? So every mechanism comes with this is counterpart. Counterpart would be if yellow cells attract blue cells. It would be pretty much same story. And the question now, when we ended up with the question, which of these mechanisms is taking place, if it's taking place during gastrulation in chick embryo. And to do this, we made simulations where we have formation of two primitive strakes in the same embryo. This from time to time is understood happens in experiments. And I have shown here 3 out of 4 mechanisms. One coincides mechanism 4 and mechanism 3 I think coincide. At the same, give same result. But so what we have here, they turn away from each other in here, these two primitive strakes. Here it's pretty much neutral. They don't, you can say they don't feel each other, they keep directions. And here they merge. And in the 4th mechanism they were also merging. And in experiments, when two primitive strakes appear, they seem to be repelling each other. So if chemotactic mechanism is explaining this, then this should be this mechanism 1, when yellow cells repelled by blue cells. Now, next part of story. In reality, cells are proliferating. And, well, adding proliferation would mean that this moving group of cells will increase in size. Well, if it increases in size and slows down, it's quite boring. What happens in reality? In reality, as we understood, for example, when this Hansen's knot moves, there are 80 cells, and there are pretty much 80 cells for a long time. And same with stem zone. There is like 1,000 cells. They move for hours. There is certainly proliferation, but this moving group is still 1,000 cells. So, in addition to proliferation, there is a differentiation. And the question is how to model it. So what we can say that again, due to proliferation, size of moving cells should increase. Due to differentiation should happen something else. For example, cells which were producing, for example, if this group of cells were producing some morphogen, then after differentiation they should stop producing. Yeah, for example, they become similar to other surrounding cells. But then if they don't stop producing proteins straight away. So there is some, for example, mRNA, for example, stopped to maintain, and it decays, and the production of protein morphogen also decays over time. So this is described here. So here, in addition to variable which was before, we add one more variable, and this variable now defines concentration of the mRNA, and then this concentration of this mRNA defines how produced our morphogen. And the question appears how we can maintain size of this group of cells, or what should be rule, or under what conditions cells should proliferate. Again, so to make everything minimal, it probably can capture even more complicated scenarios. We can assume that if concentration of this morphogen very high, cells differentiate. So this is threshold, for example. So if concentration of morphogen above this threshold, then these cells stop. In these cells, like level of production of protein starts to decay, so they differentiate. And this way, size of this segment can stay constant. And when we checked this, we checked this in simulations rather than analytically, we found that size of this group of cells can be stationary, so it can just stay, or it can oscillate. It depends on model parameters. I will show oscillating behavior. So this is group of cells. It is in co-moving frame of reference. So when it gets smaller, concentration of morphogen increases. When it increases, this while it's this segment is increasing because of proliferation, but when concentration is too high, it shrinks again. So there is somewhere appears point where inside of this segment where a concentration is too high, and then part of this segment, so cells in part in the tail of this segment differentiate, and then it gets again small, and since it gets small, there is again low production of protein, and it starts to grow again. So this kind of oscillations. But there is a domain where it's only stationary. When there are stationary solutions, so it can just fix size of moving segment. There is a proliferation and there is a differentiation, and rate of proliferation equals to rate of differentiation. It just stays same size. So this how it looks like in cell reports model. So this group of cells, they move chemotactically and they produce chemical. But if concentration of chemical too high, they differentiate. These green cells are differentiated cells. And these green cells, they don't move, they don't produce anything, they're just stationary. The question with this was that according to experiments, cells stop when concentration, for example, of FGF8. So here I would refer to FGF8, as I said before. When it's rather getting down. To model this scenario, we had to introduce one more differentiation event. So this again, when concentration is too high, first differentiation takes place. These cells still produce chemotactic agent, but they don't produce chemotactic agent, but they move, and they stop moving when it's already low. Yeah. So this, at least, it's some kind of set of assumptions which doesn't contradict to experimental observations. Cells stop when concentration of FGF8 low, but this means that we need cascade of differentiation of cells. Okay, so now it's about conclusions about this. So we have checked and found that homogeneous group of cells can move, and there are 4 scenarios when it can move. This self-repelling group of cells, it was reported by a few people, So there were a few publications, but there are 3 more scenarios when this group of cells can move. It's even these cells forming this group are attracted by surrounding cells. Or it's even more interesting cases when chemotactic activity is exhibited by surrounding cells, and this group seemingly passive, but it still moves. For example, when surrounding cells produce chemoattractant for themselves, since it's surrounded by this kind of chemical and this kind of action, this group of cells moves. It's very counterintuitive. With inhomogeneous group of cells, there are a few scenarios, or at least in 1D case, they can be analytically treated. For 2D case, they can be verified on cellular pods model. And the question, of course, whether it has anything to do with what is going on in real life. So there are no experimental verifications that, for example, during gastrulation, cells in the epiblast move chemotactically. I mean, what he checked that FGF8 and FGF4 have chemotactic, so they chemotactic agents, but it was checked with mesenchyme cells. With cells on epithelium, now it's not quite clear. So the difficulty there is that in epithelium they have properties very different from like crawling cells. It's like connected tissue, and cells move in a tissue. So the only way to see how chemotactic, how it could be chemotactic, is to assume that if cell gets signal instead of making all this machinery which is done by amoeba, by crawling cell, it does something different. For example, when signal in the front, then instead of moving, it just shrinks to sides. By doing so, this cell doesn't move, but it gets cells behind closer to source of signal, for example. I don't know. But this, what was presented here, might be much more relevant to motion of metastatic cells for cancer. So for cancer, it's definitely they move chemotactically and they move in tissues. There are chemotactic cells which move on epiblast, on epithelium. So therefore, it could be much more relevant to cancer than to gastrulation. Okay, so I was collaborating, and I'm still collaborating, and with Keith Weyer on this work, and in some part we were collaborating with Ruth del Corral. She's now in Madrid. Okay.
The earlier pictures you showed, the motion of the collections of cells, the yellow and the blue, in the surrounding red medium, are very reminiscent of what physicists call Janus particles. These are small spheres in just a viscous fluid, but the half of the surface of the sphere produces a chemical and the other half does not. A gradient in that chemical produces, near the boundary, produces a tangential pressure gradient, which by a process called diffusiophoresis, and that pressure gradient causes the sphere to move. And there's quite a lot of work in the physics and fluid mechanics literature on these things. Now, it's only an analogy with what you've got, but in a sense, it's rather similar, at least to those early parts of what you talked about. It's quite interesting.
Could be relevant to many as well. I talk about chemotaxis.
And these, by the way, these Janus particles, they can be made, and the theories and the experiments are closely matched, which I get the impression you haven't quite got to that point with the cell collections.
Mm-hmm. When I model big group of cells, they make like this kind of umbrella shape when due to self-repulsion. and it's similar to the shapes of cells like fibroblasts, yeah, I believe they called, which make this kind of shapes. It's again, so if you look into details, it could be very different, but it's possible that you end up with same kind of equations.
And the flow patterns would be similar.
Could be,
So I have two questions. The first one is, you have these number of scenarios, but you haven't said anything about direction or directionality. Do you get the same directionality of this group of cells in these inhomogeneous cases for the differences of attraction versus repelling? And the second question is, I'm assuming that the cluster that's following this other group, have you tinkered with maybe only some of the cells can read that signal and the others can't, and what might be having them move with the population?
Well, to the second, I think it was part of the model when part of cells in a group was reacting and part was passive. Here, I think here. So there is some chemical which is surrounding. We have group of cells where there is no production, but only part of these cells are reacting chemotactically. So this is, I don't know. So I was considering this as a pretty much like mathematical game. If it's relevant to something, it would be very good. About directions, it's again, so it should be pretty much straightforward. If we consider a heterogeneous group of cells, then everything is symmetric. If, for example, certain subgroup is on the right and another on the left, and when you reverse it, then everything should be like mirrored, yeah, all results. But well, for every single case, we should really consider what is geometry. For given geometry, there could be difference between right and left. When everything is symmetrical, then there is no difference between right and left. More interesting question is what happens when we have group of cells in 2D. In 2D, okay, it randomly picks up direction and moves at one hand. On the other hand, there is angular instability. It can turn. And if it turns, and if you look for long enough, it will be random motion. So it depends. If its chemotaxis is very strong, and we look for short time, we see directional motion. But if chemotaxis is relatively weak, or if we're interested in long time scenarios, then it could be, and medium is big enough for this motion, then it eventually can look like Brownian motion.
So since the primitive streak has to undergo this really long distance migration, then would you predict that an attraction or repulsion would give you higher direction or persistence over a longer time, or not?
I think persistence most likely is conditioned by something else. There is some kind of loss of cells from two sides, or there is maybe something else. So there is something which stabilizes this motion. Most likely it's not related to chemotaxis. I don't know, maybe mechanical tension, or I don't know. So maybe there is something which keeps what I mean. In this case, it's like 1D case. It can, depending on initial conditions, it can move right or left. But if there is some kind of preference to stay on the same line, then it will stay on this line.
Okay, we have no more questions. Thank you.